participants for the fourth day of the evaluated course. And we are very happy that all the sessions are lively because the participants are very interacting during the sessions. So today we are having two sessions. The first session is on gene prediction methods using Genscan by Dr. S. Vanitha ma'am. And the second session is on insilico PCR and primer designing by Dr. S. Padma ma'am. So now I request Dr. S. Vanitha ma'am to start the first session. Ma'am, please.
Thank you, ma'am. Good evening to all. So today I find the participant number as 30. Slowly, maybe it may increase. So before that, briefly, we will try to understand about this particular session, 1 hour. So actually, the content what we have designed for this day is gene prediction. There are 3 different tools for that one. One is Genscan, Grailer, Gimmer. So 3 different tools are there. But the very easier one to run the tool as well as to get the output and interpret is comfortable and easy with the Genscan. So you can try if Gimmer and Grailer tools are also available. They are a little bit difficult to open the site and get the data quickly. So Genscan is easy. So we will see about how to use the Genscan. So for half a session, I'll be dealing with this one. So to take you more interesting aspect related to the genes and the prediction of overlapping genes, we will show you using Galaxy how to identify the overlapping regions or genes present on the chromosomes. So to start with, first let me start with the gene prediction by using Genscan. So I hope I am sharing my screen. You are able to see it. Just say yes and we will start. Is the screen visible?
Yes, ma'am.
Am I audible?
Yes, ma'am, you are.
Yeah, thank you so much. So for today, we are going to see about the topic prediction of a gene. So I have categorized that as intron and exon using Genscan. So since we have a different diverse population among the participants, because some of them I find them as first years, I don't know how far they know about an intron and an exon. So for them, let me tell you that whenever you take a coding gene, which will be expressing a protein, that coding gene in that is at mRNA will be having the introns and the exons. So these introns are the intervening sequences, or we call them as the non-coding sequences, and exons are the coding sequences. So what generally happens during the processing or the post-transcriptional modifications of mRNA is that introns will be spliced, removed, and the exons will join very much systematically with exon 1, with exon 2 and exon 3 and so on to get the mature functional mRNA. So that this mature mRNA, when it goes into the cytoplasm, translation will take place and the biologically functional protein will get synthesized. So that is what we this intron and exon means. So coming to the tool which we can use to locate the genes and the exons and introns, where it will start, where it will end, all those things. We have a bioinformatic tool called as the Genscan, and it will predict the location of genes and their exon-intron boundaries on the genomic DNA. Next point, it can predict complete gene structure that includes exons, that is the coding sequences, introns, non-coding sequences, and whenever we want to do your transcription under gene expression, we have transcription, translation. So if a gene has to undergo transcription, we require the promoters. So promoters are the regions where the RNA polymerase will bind and initiate the process of transcription. And along with that one, we are also able to predict the position of your terminator signal regions. That is, here we have written it as polyadenylation signals. So this is what we are trying to predict with this particular tool. Now, what is the background program that is running behind to for the Genscan to scan the genes and predict the position of your exon, intron, where it will start, where it will be ending, and where internal regions where it is, or in promoter polyadenylation site where it is. The tool is the generalized hidden Markov model, which will be running behind. It uses the model parameters that account for difference in the gene density and the structure across the human genome. So this tool was developed by Christopher Burgee. Now, what is the prediction you can do with this Genscan? First one, position, length, and the reading frames of your predicted exons. Second one, the final predicted protein or the peptide sequence also you can obtain. You are giving a gene. You are trying to put a gene into the tool. So it will, based on the exon, intron positions, what the tool has predicted, it can give you a predicted peptide or a protein molecule. Then corresponding spliced DNA sequence also you can get with this. So this is just a brief introduction of the tool. So now I'm going to further show you how the tool looks like. All the PPT will be like that. Then I'll open the Google and show you how to run the Genscan. So if you are working with your systems, you can open it, and in between you can see what I'm giving you. You can start to work also. So this is Genscan web server at MIT. Now when you click Genscan in your Microsoft Edge, you please use, because this site is not opening in Google. So if you use Microsoft Edge and that location, I think this site will open up. Otherwise, I'll give you the link when I come to the last slide. Okay. So now here there are some, this is how the front page of your Genscan web server tool looks like. So organism here is vertebrate, which I have taken the picture as. So you can see the scroll down button. So if you do that one, you can look for the other organisms present. Then the next one is suboptimal exon cutoff. So here it can range from 0.1 lower limit up to the upper limit. The upper limit what they have given here is 1. So you're taking a gene, you can try to change the cutoff. This is maximum. You have the minimum also. In between, more number of cutoff values are there. So you can take one single gene, you try to change the exon cutoff value and see which and all prediction happens, and you can compare and see what is the region. Is there any change when you change the suboptimal exon cutoff? Then it's asking you to give the sequence name. If you have it, if you want to, you can load into it. Okay. The next one, what you find is, so I have given you here. So you can see the scroll down button. You have the list of all the cutoff values. So to start with, I'll do it as 0.05. When you're working with, maybe take one you take it as the lower end, other you take it as the upper limit, and check out what is happening. Okay. So in the print options, you can take any of these, and you can see a small window which is opened up here, clear white it is. So this is the place where you are going to paste your DNA sequence. So you take a gene from NCBI or from EMBL or from DDBJ, and you can paste your DNA sequence. Okay. After that, you can, after pasting it, now you can retrieve your nucleotide sequence from the database as FASTA format. But when you're pasting the DNA sequence here, maybe just the basis, if you take it and paste it also, the output will be obtained. So just after pasting the sequence, just you click the Run Genscan. So this is the Genscan output. First page, it will be looking like this. So you can see the date in which I have run it, how much time, etc. And you can see some of the information here. The sequence what I have taken is 8,750 base pairs, and this is giving you the G plus C content. So whenever you take a gene, identifying the number of guanine and the cytosine and finding the percentage will allow us to tell about the melting temperature with which you can determine what could be the optimum temperature to melt the DNA. Not only that one, when you have high percentage of C plus G, that also gives about the stable nature of your DNA. Then you have the ISO score 3, which is also related to the G plus C content. And when they have high amount of G plus C content, that means it is G plus C genes. If it contains A plus T content, then you call it as A plus T genes. Just the tool predicts it and gives you the percentages. Now you just come below, you can see certain regions over here. So this is all the letters or the symbols which you have to now look into it to understand what they are. So under each and every one, you can see some terminologies over here. This is what we are going to learn now. So you have, this is S stands for the strand. Now here you have the type. So here you can see INIT, that means initiation. Okay. Then you have INTR, internal. TERM, terminal. Then I told you polyadenylation, PLYA. So these are telling you the type of the regions which you can observe. Then this is beginning. When I give the gene 8750, from where the initiation can start and where it will end. It is predicted. Then what could be the length from beginning to the end? And this is FR, this is pH. I will give you all these things later. Then I by AC, initiation, and then this is DO by T, coding regions, and also the percentages. So I'll give you the interpretation of the results shortly. And the same extension of the same one, little more details about the same thing it is giving you. Okay. So I told you that it's not only giving you the position, beginning or ending of an exon and an intron, but it is also giving you the predicted peptide sequence. So you can see that for the gene, what I have given as an input, this is the predicted polypeptide chain from it. Now you look into this one, which is explaining to you about each and every parameter that you observed as a result on the output of your Genscan. First one, what it was, GN and EX. What does that stand for? GN stands for gene number. EX stands for the exon number. Okay, so INIT stands for the initial exon, that is initial, whatever is the beginning of it. So ATG towards the 5' splice site. Then INTR stands for internal exon with 3' splice site to the 5' splice site. Then you have TERM. These are the terms what you find in the output. You have the terminal exon, 3' splice site to the stop codon. Then SNGL stands for single exon gene, ATG to the stop region. Then you have PROM, which is the promoter. So if you know about, if you have read all these things, you must have seen all these terminologies in your molecular biology. You call it a startup box or the initiation site, and PLYA indicates the poly A signal, which will be having the consensus sequence AA, TA, AA. Okay. Now, furthermore, there are more other words given over there, right? So what are they? One you have seen, yes, I told S means a strand. So whether the strand is positive strand or it is the negative strand, opposite strand. Then BEGN is beginning of an exon or a signal. Whereas END stands for what? End point of an exon or a signal. LEN says what? It is a length. So from the beginning to the end, if you subtract, you will get the length of your exon or the signal, whatever is the output. FR is called as what? Reading frame. Whereas pH is called as a net phase of an exon. I by AC is called as initiation signal, or you call it as the acceptor splice site. DOT stands for donor splice site, or the 5' splice site, or the termination signal scores. Then CODRG is called as coding region score. P is called as probability of the exon. So sum of all this is actually like running with your parsers. That means it is some algorithms to which will be containing the exons, and then TSCR stands for the exon score. Okay, which is dependent on the length, then I by AC, DO by T, as well as by the coding scores. So with all these things, we'll be able to finally get into the exon score. So these are the mathematical calculations which will be done in the tool. Now, once you do this one, you look into one of your column which is having the predicted donor splice site value, which should be, if it is more than 100, then it is an excellent region to be considered as an exon. If the predicted percentage is between 50 to 100, it is okay, acceptable. If the value is between 0 to 50, then it is very weak. It's very difficult to consider that it could be an exon, and any value below 0, because some negatives also may come. So if it is below 0, it is a poor value, probably not a real donor site. Okay, so now I have taken one sequence, same thing. I have taken the FASTA format and run the Genscan for this particular gene. So let me show you the same result. Now you try to understand the terminologies which are present over here. What is GN stands for? What does GN stands for? Anyone?
Gene number.
Yeah, EX stands for?
Exon number.
Exon number.
Yeah, type stands for what? Whether it's an initiator, initial signal, or the internal regions, or the terminal regions. S stands for what? The type of a strand. Okay, beginning, ending. What is this? LEN stands for? Length. Okay, then FR stands for the number of possible reading frames. Then what is I by AC?
Initiation signal.
So DOT stands for what?
Donor.
Donor splice
Donor
site.
splice site.
Yeah, so this is what I told you. Have to check the score what you got. Now in this output, when you take the DOT, you just check out the values over here. Did you get negative here? You got your negative. That means it is a very poor region to predict presence of any exon regions over there. Now here you had 110, here you're having 109, here you're having 119. So these are the regions where the prediction is very good that the possible presence of exons is in these regions in that particular sequence, it is very good. So you have some values in 94, you have something in 47 also. So depending on this percentage, you can predict whether that particular region what you have considered is having a exon possibility or so. Okay. Now the same thing, more version about it. You can see here I have taken the suboptimal exon level is 0.010, and for this sequence you can see the peptide also. I have got it as the predicted peptide sequence as the output. Okay, so now this is the site actually. So I am trying to do here, just check out whether you are able to log into it. I'll post this one in your chat box, this link, and see whether you are able to run it, or if you can do it in using your, just typing Genscan in your thing also, just to see whether you are able to. I have posted it in the chat box, see whether you are able to, go into the site. Anyone has tried?
Trying it, ma'am.
Yeah. So I have opened the site. I have just opened it now. For me, it has opened like this should be there. It is opening in Microsoft Edge. So if you take this link also, Google, I don't know how far it will work out, but you can try it out in your, okay. So now what we will do is that same for procedure, we will try to go to, somebody was asking me about insulin gene previously that they are not able to. So let us see why it is. So I'm going to NCBI. So it's National Center for Biotechnology Information. So here, As per the request, I'm putting the gene. Okay. I am taking insulin. Okay. I'm giving the word as search. Okay, so now I got the list of genes, but here it is insulin receptor because I told you that the first page which is opened up will be having items from 1 to 20. But you see how much data is related to insulin, 91,000 are there. So if you want insulin gene of Homo sapiens alone, but here you find Homo sapiens only, but it is like receptor, growth factor, and so on. So what the individual who had asked me the question is go to page next. You have always an option to go for the next page, right? So now the next page is open. Now here I am able to get the insulin gene of Homo sapiens. So now you can click this one. So if I click the gene database will get opened up. So you can see the gene ID, the details are given here. Now, as you go down little bit below, you have here the FASTA. So just click the FASTA format. You will be getting the sequence downloaded. So you can see here Homo sapiens for insulin. I got it. So now what we have to do is take only the sequences, copy it, and we will go to the Genscan. So cutoff at present, I'll take 0.05. You can just try with the different parameters and check with one or two what is the difference. So just you can say run Genscan. So within a few seconds, I got the output. I hope if anybody's working out, you'll be able to get it. So the number of gene bases for insulin is 1431. And you can see the G plus C content, it is 64.64%. Okay. So now you can look it into the all parameters over here. So there are only 1, 2, 3 regions which are being predicted. One is the initiator region starting between 239 to 425. And you can see the predicted donor splice site percentage is 84, whereas the terminal sequence, it is only 42%. And you have the polyadenylation, not much good percentages are being observed here. And when you go just below there, you can see the internal exons also. So you can see the score is 84. So now with this gene, what we have loaded, you can see the polypeptide also obtained over here. Okay. So this is how you can use this particular tool to approximately predict the position of your initiation region, terminal region, and also the internal exons, whatever is possible, it is able to predict and give. So now you can do the same thing with any other gene also and try to check out what is your output. Is anyone trying it out?
Yes, ma'am.
Is this link was working for you?
Yes, ma'am.
Okay. So have you done with this one? So this is over. If any doubts you have, you can ask me. So otherwise, we will go with the next one. Yes, all of you are working with me. Yes, eyes wide open.
Yes, ma'am.
Yes, ma'am.
Yeah. Others can put Y, capital Y in the chat box. I can just have a look. No doubts means small n, small d. Okay.
Ma'am,
Yes.
I can't use the chat box, ma'am. It's showing that you can't send message because you're not a member of the chat.
Okay. Okay. You are.
I've
You can
messaged it
unmute and
to
speak,
Padma,
right? So.
ma'am.
Yeah, you can unmute and speak so that we are happy to hear
Okay,
your
ma'am.
voice and get the information from you. That's okay for us. Okay. And
Okay.
what is the applications of this Genscan, ma'am? I showed you the PPT first slide. Where were you? What are the what you can predict or what is the uses is position, I'm sharing the screen, right? Position, length, and reading frames of your predicted exons, and then the final predicted protein and the peptide sequence can be predicted, and correspondingly spliced DNA sequence also can be predicted from the Genscan output. Whoever has asked, I hope I have answered your query. Okay, so shall we go to the next content? Okay, so now what we have seen is we have just tried to look into the Genscan tool to check out and predict the position of exons and the other things. So now what we will do is we'll go to the next session on the topic, which is visualizing the overlapping genes using Galaxy. I would like to go a little bit faster because this will take some time in the practical part of it, because we'll use the Galaxy over here. Okay, so what are these overlapping genes? So short form is given as OLGS, which are adjacent DNA segments that overlap with each other, producing two different proteins. Okay, they're also known as dual coding genes. How do they work? They are translated into two different reading frames. One is like this, the other one will be like this. So that will result in production of two different proteins. And any mutation on the DNA overlapping region will affect the transcription of all the genes which are involved in that. And one of the important understanding about this overlapping genes is which is important in regulating gene expression. Now, where can you find these overlapping genes? In the prokaryotes, even in the eukaryotes. And some example for you to understand what I am trying to talk to you about is like examples. The pictures are telling you where possibly the overlapping can be when you take the two genes. Here you can see that you have this is your 5' end, 3' end. This is your 5' end, 3' end. So at the 3' end, there is some overlapping between the two DNA strands. So this is called as convergent. This is divergent. This is parallel. You see the arrow mark, and based on that you can try to understand whether they are parallel or whether they are anti-parallel. And you can see the position where this is your 5' region, this is your 3'. Okay, so like this, there are some partial overlaps. There are also some complete overlapping regions. So by using Galaxy and different set of tools, we will try to predict in a particular chromosome, is there any overlapping genes present within them. Okay, so for that we'll directly use only the Galaxy tool for today. So you, as usual, you open the Galaxy tool. If you are very fast enough, you can please run it also. So you just open the Galaxy tool and then you are going to create a new history. That's how I thought it becomes easy for us, because you have to rename the process when it is running, so that it becomes easy for you to select it when you are running the different other tools. So in that history, you can rename it as Genomics Galaxy Genes or whatever you want to know. I have renamed here. You can rename your history also and click the save option. After that, go to your tools section in the Galaxy, and there you can type UCSC. UCSC stands for University of California, Santa Cruz. That's an main table browser. You have the main page of UCSC also. If you go there, there are a list of tools available. But in that list of tools available, we have to use only the main table browser. So your Galaxy tool will directly give you this UCSC main table browser. So once you select this one, automatically the Galaxy will take you to the home page of your UCSC main table browser. Directly it will take to you, and this is how the page will look like. Now there are some set of parameters what it will ask you to select and do it. So what you do is that this select data set is there, that you don't do anything. You just keep all the parameters as such, but come to this define region of interest. In this region, you have the region as genome and position. Now you select the position and already when you open the tool, there will be some chromosome number given. But for our exercise, you please take chromosome 22 because it is the smallest or the shortest chromosome. So let us see what our prediction is going to be. So just put chromosome 22 and then no other parameters to be set. You will just go on. One observation you can make here that it will be asking you in what format you want the output on the list of genes that are present on chromosome 22. So it's called as BED file, BED. So you can see that BED browser extensible data. It's already by default it will be there. You don't have to do anything and just you say it as get the output. So the moment you click get output, you will be having the next small window appearing where the output will be coming. Here also don't do any changes. You just make sure that under create one bed record per, you just see that it is selected as whole gene, and here it will be 200 number, and you can just see down, no other parameters to be done anything. You just go down and see here the small term given here, send query to Galaxy. So if you just click this one directly, the data will be transported to your Galaxy. So you can see that when I clicked that one, you can see on to my right hand side, the data has got deposited in my history. So history name is Genomics Galaxy Genes, and you can see the chromosome number 22 and all the relevant details in what format I said to you, in the form of BED format, it will get deposited here. Okay. So now what you do is that you want to expand and see what is there in a row, what is there in a column. So you can just click it. You have the eye here, right? Preview. You can just click that button here and you can see that whatever you have in the small window will get expanded. So you have the set of here the columns and you'll be having the row. So if you see the row, everything we have taken the genes that are present in chromosome 22 only, right? So it is telling starting, ending, and what is that gene name, and then it is giving you the scores. So now this is strand. So now if you observe this one, this is column number 6. Always remember, we are going to check the genes, overlapping genes. So what we require is the strand. Now other informations, maybe if you are looking into it for understanding, you can have a look at it. But for today, to check the overlapping genes, you have to see only column number 6. I'm insisting that word, column number 6. Okay. So now this is 1, 2, 3, 4, 5, 6. Now you can see some are minus, some are plus. So what does that indicate? Plus strand indicates it is a forward strand. Minus indicates that it is a reverse strand. Okay. So the data what you have got from UCSC has given you the column 6, which is indicating whether that gene is present on the positive, forward, or in the negative strand. So it is a pooled up data of both the strands. Now what we are supposed to do, we are supposed to separate the positive strand from the negative strand. So you have to filter the data. So for that filtration, you have to go to your tools department again. There you have to select one. Just type filter over here. There you can see filter data on any column using simple expression. Select only this, because if you type just the word filter, lot of information related to filter will be coming out, but you make sure that you select only filter the data on any column using simple expression. After that, when you come to the middle department where you have the tool parameters to be set, there you can see that in the filter, make sure that this one is your genes on chromosome 22. Now, if anybody is working in your Galaxy, it will not come as genes on chromosome 22. It will come as UCSC gene or something like that. What I have done is that I have renamed the file over here. So that it becomes easy when I am going to take the data and merge it later. So that is why I have renamed it it as genes on chromosome 22. Okay. Then what, just come down below. Here it is asking with following condition what it is. Now, when you are clicking into your, when your galaxy, it will show C1. So that means it is column 1, but we don't want any work to be done with column 1, but we want to be do the work only with column 6. So that's why you remove that 1 and type it over there 6, and then is equal to, then it will be given chromosome 22. So there you remove and put only plus symbol. That means you are trying to filter from the column only the forward strand. Okay. Then what you do, just click on the run tool over here. Then you will be immediately getting your output. So what did you do till now? You have filtered the column number 6, taking up only the plus strand, that is a forward strand. So I have renamed that one. You can see on to my right hand side, genes on chromosome 22, the green color are there, right? So you can see, and above you have the forward strand. Now, if you, then what you do again, you have taken from your gene set, you have taken only the plus strand. We have not taken at the negative strands, right? So again, you go to the tool, again in the filter, you click filter data on any column using simple expression. Process remains the same. Now, in your filter department, you make sure that you're taking only genes on chromosome 22. Okay. When you're finishing your first filter, when you take your second filter, automatically in the filter, it will show you your second filtered data. Okay, so you please make sure that you remove that one, take up only the genes on chromosome 22, the first data set what you have retrieved from your UCSC site. Okay, then what you do, you have taken the forward strand. Now we require the reverse strand. So again, in your following conditions, you remove that column 1, change to column 6 and put minus symbol over there. That means you are now filtering the data only for your negative strand. When you run the tool, then you can see that you will be getting the output. You can see to my right hand side, 3 data set has been obtained. One is the genes on chromosome 22, green color. You can observe the green color. Then your forward and the reverse strand. I have renamed everything, children. Okay. Now what did we have done? To which level we have gone? We filtered it. You have separated from the chromosome plus and minus to 2 different data sets. One is the forward, one is the reverse. Now what we are supposed to do? We have supposed to intersect one above the other. First forward with reverse, then you have to do reverse with forward. So you have to align in 2 different ways. First one is you have to intersect. So after filter, it is intersecting. Intersecting what? Forward strand with your reverse strand. Next one is reverse strand with your forward strand. Okay. So for that one, again, you go back to your tools and click the word as intersect. There, you have to select only intersect the intervals of 2 data set. So once you click that one, again in your tool parameters, list of data will be asked. So you have to select the parameters. Now, what did I tell you? First intersection you have to do. Here you can see off, OF off is there. There you have to select what? Forward strand. So whatever you have renamed, you can use it, or just directly the second result what you have got, you make sure that it is taken as forward strand. Then what you do? Now below that, when you have that intersect, there you select the reverse strand. So first set of intersection is done between forward strand with the reverse strand. Then you just click the tool button, that is run. So it will try to locate the regions on forward and the reverse and try to see whether there are any overlapping regions between forward and the reverse. Next, you are going to do the other way around. Again, go to intersect tool, intersect the intervals of 2 data sets. Now you change it, the reverse with the forward. You have to see. That is why I told you it will give a different one. You have to scroll down the button and select only the selected data. Here it is forward. Okay. Then again, you put the tool as run. Okay, now you can see on to my right hand side here, green color, genes on chromosome 22, forward strand, reverse strand, intersect forward to reverse. Now it is intersect reverse to forward. So now if you do that one, you will now get you have now made the data set separate, positive, negative. Try to intersect to check out whether there are any overlappings between them. Okay. Now what has happened? Forward to reverse, one data set is there. Reverse to forward, another data set is there. Now these two data set has to be merged together so that you will be trying to together check is there any overlapping regions between the whole on the all the genes that are present on the chromosome 22. Okay, so for that one you have to use a tool. So again go to tool and type there as concatenate. Here when you type it, there will be list of concatenate informations coming out, tools coming out. So you have to select only concatenate multiple data sets tail to head. Okay, so select only this parameter. After selecting it, what you're supposed to do, come to the tool parameters. There, please be very careful to select the concatenate data set. Now it is asking you which two intersected data you have to compare and check it out. So there what you do is that you had your intersect data reversed on forward, forward on reverse. I have renamed all of them. So if you can, you do it. Otherwise, whatever you get 1, 2, 3, 4, 5, accordingly you take your input data for the concatenate data set. Okay. Once you take all the two things together, if you just put run the tool, then the output will be obtained. Okay. So now when we were running this particular tool, is anyone running the tool? Okay, I'll just finish the theory part. So when you are putting concatenate with all these positive, negative strands intersect regions, we can observe that approximately 4,000 regions are showing that there are overlapping. Okay, but practically or logically, when you take so many number of overlapping regions on the genes, that is not actually acceptable information, because that's what you get in your concatenate regions. If you are clicking that one, you'll be able to see it, how many regions are overlapping. So what we can do is that now you can screen down the data set so that you will be getting only the correct overlapping regions. Okay, so for that one, what we can use is that you have a tool in your galaxy which is called as gene bed to exon intron codon bed. So this tool will try to extract only the exons present on the genes of your chromosome 22. Earlier, what did we do? The whole gene we were trying to check the position of overlappings, but we wanted to see only on the exons. If you want to do with introns also, no problem, because in this bed program, you have some extract data over here where I have selected only coding exons. You can, if you click that one, you'll be getting introns only or exon introns like that. Lot of filtering will be there. So you have to select what you want the output to be. So for our understanding, to check the overlapping regions on the exons, we have selected only the coding exons. Okay. So now what you do, you take only the extract data as coding exon only. Now the next one is from where you have to get your exons region only. Now here you please be careful to select only the first set of data what you have retrieved from your UCSC. Okay, you got your UCSC data set. From there you filtered, intersect, concatenate. One workflow is over. Now you want to little bit change your workflow. Instead of taking the whole set of genes in the chromosome, we are now screwing down. We are now scaling it down. So instead of taking the whole gene, you are taking only the exon or the coding genes. Okay. So for that one, again, your workflow will start from where? From your beginner. Who is the beginner over here? UCSC genome table browser. From there, whatever is the chromosome 22 data, what you have downloaded. Okay. So start from there. And so you have to select here that only. And when you put the tool as run tool, it will run and it will be giving you the output. Okay. Now what you're supposed to do now is that your workflow now onwards will be the same. Filter, intersect, concatenate. So if you repeat all the steps what I told you from 7 to 11, you will be getting out the parameters output. So I have taken it on chromosome 22 and this is what is the output result. I have got it out. So when I'm using the initial process of taking all the genes present on chromosome 22, the overlapping regions were found to be as 4,423. But when I use the bed exon intron tool to scale down the regions where the exons are having overlappings, then the number has got drastically reduced to 11 regions only. So now this is a parameter where the overlapping regions on chromosome 22, initially I got with the whole data set as 4423, but with the help of this bed data tool, I have shortlisted only the 11 regions within the chromosome number 22, only on the exon regions. Okay, so if you are able to run it and get it, I'll be happy. And this in the same thing, Galaxy only, you have something called as an history workflow. I did not do anything. I just did the process yesterday and I got all the workflow what I did. Okay, so here you see I have renamed chromosome genes on chromosome 22, filtered them, then intersected them, then I got the concatenate data set. Then what did I do? Since the data set was showing more number of the overlapping genes, I went back again to my genes chromosome 22 and I filtered to get only what data set by using the bed exon intron codon tool to get only the exon regions. Then what did I do? Exon regions, again I filtered on the positive strand, negative strand, forward, reverse, and then they intersected between them. After intersection, I made them to undergo concatenate. Okay, so that's how I got the workflow, also the output of this protocol. Okay, so that is how you can use your this tool, that is Galaxy, to predict the overlapping regions or within any genes it is there. Okay, so if you understood till here, just put one Y, capital Y in the chat box. Or I was very fast. Anyone is responding? Okay, so I think today it is very less Y only. So what I'll do is I just opened the Galaxy tool. So I'm left out with some 10 minutes or so. So just initially I will just show you how it was being working on. What are we supposed to do? I told you first one is what? You have your unnamed history, right? I am just, you rename it as what? Whatever you want to rename it as, you rename it. Just whatever is possible within this time, I will tell you. Just you rename it. It is only for having the history, okay? And just put save option. So whatever data I get will be within this genomic Galaxy gene, okay? What is step number 1 you're supposed to do? UCSC main table browser, okay? So you can see here you have the select data set, don't do anything, but here in this defined region of interest position, you can just take chromosome 22 alone, just click only use that one, then retrieve display bed tool it is and get the output. So if you click get output, as I told, the window will open up where it is whole gene 200 bases upstream and downstream is still 200 bases. Now I'm asking it to send the query to Galaxy. Okay. So now you can see that onto my right hand side, it is loading the history. So it is still in the process of doing it. So it has started, maybe another few seconds, it may be coming out. Okay. So here only I told you can rename it. So here you have the pencil symbol, right? So that if you click, it will ask you to edit. So there you can rename it as genes on. So it's turned green. So I've got the output. Okay. So I've got here, right? So what you do is you just copy this or cut it and paste it in the info, and here you can name it as genes Okay, I already have it. So I'm just doing this and putting the word as save. Okay, so now you can see it has been renamed as genes on chromosome 22. Okay, so now you can open the thing and you can see it is showing you 12 columns. But here I want column number 6 only. So if you click the I icon button, you can see 1, 2, 3, 4, 5, 6, 7, 8. So many columns are there. But let us check out the column strand. Here you can see sixth column, it is having positive and the negative strands. Okay, so now what we are supposed to do, we are supposed to filter the data. So what is the tool you will be typing is filter. In this one, which one I asked you to take? Filter the data on any columns using the simple expression. So if I click that one, again you can see automatically in the filter, since onto the history I have only the first data, that is genes of chromosome 22, it has taken. But with following condition, it is showing you what column 1 it is showing, but I don't want to do any filter with my column 1. I want to do filtration only in which column number 6. So you put here as less. Okay. What you're trying to do is to retrieve or take the filter the data which will give only the data for forward strand. Then you can run the tool. So you will be getting the output which will be giving only the positive strand data. Okay. So here again you can rename it. So what you can do is that you can put the pencil button and rename it as forward strand. Okay. Next, what you will do? Again, go to filter. Now you have to filter what? Do you want to filter data 1? No, I don't want that one, right? So you put the scroll down button and you have to select now genes on chromosome 22. You don't have to do anything with your filter data 1. You have to do only the work with your genes on chromosome 22. Again, here we want only for the which strand? Minus strand. So I've taken minus and I've said run the tool. Okay. So already I've got the filter data on 1. Which strand it is? Which strand it is? All are on mute. Okay. Forward strand. So you can just save it. So it has been renamed. So what is the second filtering you're doing is which tool you're filtering it is your
Reverse strand.
Reverse strand. So you can rename that one also as the reverse strand. After this, what are we supposed to do? Anybody has got the result output for first intersection and then the first concatenate when you're joining it? Anyone has got? Okay. After filtering, what we are supposed to do is what? Intersect. Yeah, intersect. Which tool you're supposed to use for intersect? You will use only the intersect, the intervals of two data sets. Okay. So when you use this particular tool, parameter is overlapping interval. So you don't have to do any over here, but you have to see what is this OF of here. What is the strand here? Automatically it is showing you reverse strand, but let us convert it into what strand now? Forward strand. So intersections between first you do with forward strand with that of your intersect with your reverse strand and you can run the tool. Okay, so this is your first intersect forward with that of your reverse. Okay, then again we will take intersect. Now what intersect you have to do is you have to do reverse with that of forward strand. Okay. Then you have to run the tool. Already this is over. Maybe we can rename it. So what you would like to rename it? Intersect only we will put, but we will mention which strand on which strand, forward on reverse. Okay, and just save it. So even the other one has finished. So this also you can rename it. So what is the intersect? Reverse on forward and you can save it. Okay, so that also is done. What is the last step you have to do for your data is concatenate. What are you supposed to take for this one? So many concatenates are there, right? So which one you're supposed to take? I told you. Which one you're supposed to take?
Tail to head.
There are so many tail to head, right? So which one you're supposed to take? Concatenate? What
Multiple
happened
data
today?
sets.
Multiple data sets. You have a concatenate multiple data sets head to tail. Okay. Take this one. Now here you should be careful to take the two data set which has to be now concatenated. Okay. So already it has taken here intersect reverse on forward. Now you have to take which one intersect forward on reverse. Okay. Then you will run the tool. So if you run the tool, you'll be getting the output. Yeah, it's working now. So you will be getting the output. So that output, you can just keep it aside. So now this is the workflow, what you have done till now, from getting the data from UCSC, and then you are going to filter it. Then you're going to intersect positive, negative, and then concatenate. Okay. So now you can see the set of your regions, which is predicting to be as 4,423. Okay. Now here I will tell you how to check your workflow. So here you can see this is the workflow. Create workflow. If I give, it will show me. So here if you check it out, it will be showing you the workflow, what I have done. What is the use of this workflow is that if you want to do on chromosome, some other chromosome, you can take the genes and you can ask the tool to run the same workflow. So you have that run button, right? So if you just run button, if you do it, then it will run the same one. So what I have done till now is your workflow, what it has coming out. Okay. So now what we will do is that we have finished till here. Now what is the next thing tool I told you you can use to, instead of going with the data what you have got from your initial stage, you now go with what tool I said? What is the tool? Yeah, tell me quickly. No one has registered in your mind. Yes, get. What is that one? Get bed, gene bed. What happened today? Very slow. Gene bed to exon intron codon bed expander. Okay. So now you can see here in the extract what it is showing you. You can see it is showing coding exons plus the untranslated exons, coding exons only, UTRs. Then you have introns and codons. Let us take only the codons, coding exon regions. Okay. Now here be very careful with from where you want to extract only the exon. So here you be careful to take and select only the genes for chromosome number 22 and say it as run the tool. So it will run the tool and you'll be getting the output. Okay. So now it is running. Now what we are supposed to do from whole gene, you are now concise to get the only the sequence or the only the exons you are now getting it out. Okay. So now from this exons, what you're going to do now? What's the next thing you're going to do? You're going to repeat the process. What is the first process? It is filter. What you're supposed to filter is data on any column using the one. So here you have to filter which one? Which one you are supposed to filter? Gene bed to exon. So run the tool. So you will get that is serial number 8. What is that strand you are getting? What did I do? Okay, so this one I will delete. So what you're supposed to do is what you have taken gene bed, but we didn't change the column, right? So what is the column you have to take is plus. Okay, then you can run the tool. So you will be getting the forward strand. Next, what you're supposed to do is take 7, that is gene bed, exon data alone, and then you have to take 6 and then go with what symbol? Minus to get your reverse strand. Okay. Then what you're supposed to do is now intersect. What you're supposed to do in intersect, which one you're supposed to intersect the intervals of two data. What you're supposed to take here is what it has taken by itself filter on data 7, that is number 10, and so this should be number 9 data set. Okay, so that is 10 and 9 serial number I've taken here. 10 and 9, so now I should take 9 upon 10. That means forward with forward that intersect on reverse and reverse that intersect on forward. Okay. So it's running the tool. Now what is the last set you have to do is concatenate. So what you're supposed to take this one is multiple data sets tail to head. Okay. So still it is running the 12th one. So let us wait for, yeah, it has finished. So now I will take intersect the data on 9, no, 10, 10 on 9 and 9 on 10 and run the tool. Hopefully you will get the output or the result. Okay. So by using Galaxy, maybe you can try it out later when we, if I've gone very fast, anyway, we'll be sharing the PPT with you. So maybe you can look into that one. Now you tell me how many regions it is showing you. Only 11 regions. Okay. So that is from my side. So thank you all. And Padma ma'am, Manju ma'am, I have finished. So any queries are there, please write and keep it somewhere. Once Padma ma'am's session is over, at the last we will take both the questions from my session as well as from Padma ma'am's session.
Thank you,
Excuse me, ma'am.
Vanitha ma'am. Thank you for the enriching session on Genscan. And now I request Dr. S. Padma ma'am to start the session. Ma'am, please.
Thank you, Manju. Am I audible?
Yes, ma'am.
Yes, ma'am.
Okay. So good evening to all the participants. Do you need a break or shall I continue? Full of
Ma'am,
galaxy
actually,
and
one
stars.
is show.
Galaxy and stars around you. Yes.
They not understand Padma ma'am. Malli cheppandi.
Are you finding a galaxy of stars around your head after going through galaxy? Yes, any comments? No comments? All are happy? Yes? Any participants respond? Are you happy or overloaded?
Yes, ma'am.
Okay. So shall we start the next session?
Yes, ma'am. Yes, ma'am.
Okay. Okay. So,
Excuse me, ma'am.
yeah.
Ma'am, is it possible for the sessions to have for 1 hour?
Sessions for what?
The time duration should be 1 hour.
No, 1 hour means we have total 30 hours for this course. We have to complete lot of topics, and 1 hour means it will be extending almost till end 15th of February, which is not possible for students or anyone. So what is the constraint to attend for 2 hours? What is the constraint? Someone asked for 5 minutes. Somebody asked for 5 minutes break. So is it for everyone or only 1 or 2 people? You can take a break and join back. You will not be missing much. Okay, so now you have seen how to identify the exon introns, how to look into the genes, what are overlapping genes, What is the problem you think you will face when overlapping genes are there? Anyone? What sort of problems you think you will face if you are overlooking the overlapping of the genes? Anyone? Hello? Please respond. Problem in protein structure. Understanding the expression pattern. So that is far more we have gone. Anything before or with not going at the level of the cellular events, not looking at the level of the protein structure, looking at the DNA sequence as such. If today's session you have seen that there is an exon intron boundary, there is a terminal region, and so many things. So if you have an overlapping exon between the genes, so what probable uh, chances are there that you are reading same sequence of the two different genes. One may be at the end of the gene 1 and that may be the beginning of the gene 2. So here in such situations, there can also be a situation where there is a gene which we call as a pseudo gene. A pseudo gene may be placed in between these.
Excuse me, ma'am. Ma'am, are you sharing the PPT? It's
No,
not
not
visible.
yet, not yet,
Not yet.
not yet.
Okay.
I didn't share the PPT as it.
Okay.
Just, yeah. So this ambiguity, or when we are looking at the sequences, there will be some amount of overlapping of sequences. Then you have the exon intron boundary, and also these some of the genes they may be having a pseudo gene in their structure. So if you have to work with these genes, we nearly need to amplify the particular set of genes, and then we need to see what can be done. And also these genes, as we have discussed previously also, there will be some replacement of some nucleotides, as like there is, which is what we call as a single nucleotide polymorphism and all. So today's session, what now the next half an hour or 45 minutes, whatever we are going to look at, that is going to be related to the identifying the specific position of the gene on the chromosome, and how to design, how to amplify that particular gene to increase or to understand what the kind of sequence is there, or what can we do with that particular gene, any SNP is there. So if you have to sequence the gene, or you need to look at the distribution of the gene nucleotides, then you have to amplify the particular gene. So today's session, what we are going to talk about is all about this. So let me just share the screen. Is the screen sharing?
Yes, ma'am.
Yeah. The screen is coming. I need to, I don't know, the problem is coming. We just share. Yeah, too many overlapping regions need to screen down to get the correct data. That is also true. And also interference. We are not going at the level of the cellular events, but we are trying to work at the bench level. What would happen if we are taking the wrong gene or trying to study with a particular gene of interest? Okay, so here I will again try to share my screen. Now is it sharing?
Yes, ma'am.
Okay. So today we are going to do something about the amplification of the gene. So amplification of the gene is, all of you are familiar with it. What we use generally for amplification of a gene is the polymerase chain reaction, which we call. And we are going to see normally, polymerase chain reaction means we are going to do it in the lab. We have a setup, we have a PCR machine, we have a set of reagents, we have a set of PCR tubes, and we have a set of experimental conditions which need to be satisfied. And then we use the primers, and we keep it for amplification. And later, once the amplification is completed, we would be visualizing them by running them on the agarose gel. Okay, so and now today we are not going to do all that, but what we will try to see here is, can we do this in silico manner? And what is this in silico approach? How it is going to give us a better result, so that we can reduce the amount of reactions or the PCR reactions which we do in a wet lab. Of course, whenever we do an in silico experiment, always we have to remember that they need to be supported by the wet lab experiments. So whatever we are trying to see on the by using our knowledge and by using the tools or by using this softwares, we are only trying to minimize the amount of wet lab work which can be done, improving thereby the accuracy of the experiment and correctness, instead of repeating the same experiment with multiple permutations combinations, which can be already done if you are able to visualize the genome, or you are able to visualize the position of the gene and what are the regions which you should select so that your gene is correctly amplified. So what regions we have to select, all these we can do by using the in silico approach. So today we are going to see some of these tools for in silico PCR, which includes the designing of the primers. So just before going into the experimental, before doing a hands-on session, first briefly, as we all understand, we are familiar with this term PCR. First, the purpose of this class, we let's quickly go through about this PCR and what is a primer and what are the necessities required for designing a primer. What is meant by designing a primer? So, in these next few minutes, we will be concentrating, or we will be looking at these aspects, and later we will be trying to amplify some gene in using the in silico tools. So polymerase chain reaction, the name itself indicates that it is a method for amplifying the DNA sequence. So we know that it uses a heat stable polymerase. It requires polymerase and primers, which are of 20 bases in length. And these bases is a complementary strand at one end of the sequence to be amplified, and one complementary strand at the other end. Okay, so the newly synthesized DNA strand can then serve as an additional template for the same primer sequences, and this sequence of reactions goes on. Successive rounds of primer annealing, strand elongation, and dissociation, all this will produce highly specific amplification of the desired sequence. And so now we know this once this amplification has been completed, then we need to take the product and we have to see the size of the product, how much it has been amplified. So PCR can also be used to detect the existence of a defined sequence in a sample. So this is some brief introduction to go about our program. So the polymerase chain reaction, this is a technique, experimentally how the polymerase chain reaction is done. So there are 3 stages, denaturation, annealing stage, and an elongation stage. This will go on for several cycles depending upon your experimental setup or the DNA which you are working with and the primers which you have used. You have to standardize the lab conditions for these parameters, how long you want to do the denaturation, 1 minute or 2 minutes, annealing temperature, annealing is for how long, elongation, and how each cycle, how many minutes it happens, and how many such cycles you would like to do, so that in every cycle there is some amount of amplification which is happening. So as you can see, this particular strand is divided and the primers are annealed here. This is called as the denaturation step, which is done at a very high temperature, and the primers which are there, these are going to anneal to this at a specified temperature, and this annealed primers are going to be converted, elongated for the 5' end and the 3' end, and this elongation, as I told you in the slide, it means that these strands which are forming, they will become the basis for the subsequent primers to get analyzed. So this cycle of denaturation, annealing, and elongation goes on, which we all know that happens in the PCR reaction. So there is also this quantitative PCR, which we all know now. After COVID, it has become a very household term, PCR reaction, PCR test, and all this has become a household name for us now. So this quantitative PCR is also called as the real-time PCR. Why we are calling it as a quantitative here? In this previous situation, when we are trying to use the PCR tools, we are using the primers and we are amplifying the sequence, which we are only looking at the final product which is forming. When we come to quantitative PCR, the term quantitation is basically you are looking at actually the change in the number of copies of the DNA, how it is changing, what is the comparison between 2 different types of sequences, or if you are comparing 2 different organisms, the amplifying the gene sequences of 2 different organisms, which one is amplifying, or whether a particular gene is present in sample 1 and absent in sample 2, that you can monitor on a real-time basis and once you do a quantitative PCR. So the quantitative PCR will be useful for doing many of these applications related to the gene expression, SNP analysis. SNP stands for single nucleotide polymorphism, pathogen detection, as I have told you, drug target validation, and also other applications. I'm not going to detail all those things, just to cover the brief idea about these types of PCR which are there. So real-time PCR and traditional PCR. So traditional PCR is the one which we are doing, which we routinely do in the lab. So it measures only the amount of accumulated product at the end of all the PCR cycles. Whereas the real-time PCR is going to quantify the amount of copies of the DNA which are produced as the amplification is going on. You can real-time monitor the change in the number of copies, whether it is increasing in a sequential manner, whether the amplification is over, or what kind of amplification is happening there. So The data is collected during quantitative PCR in the exponential growth phase, and when quantity of PCR product is directly proportional to the amount of the template or amount of DNA which you are adding. Whereas the traditional PCR, this is only comparing the intensity of the amplified band on a gel to standards of known concentration, and it will give you only a semi-quantitative result, and there could be some experimental errors also with the addition of the primers or with addition of some reagents. If there is any dilution problem, then the product which is formed may be of not high intensity. So in that case, we cannot say that because intensity is less, there is no amplification. So there is many chance of any experimental error coming up there. So what are the disadvantages of traditional PCR? The poor precision, sensitivity is low, and the range is short dynamic range, low resolution. It is not automated. It is based only on the size. Then there are no number of amplicons which are formed. That information is not there. Ethidium bromide, which is used for staining agarose gel to visualize the PCR product, is not a quantitative method of visualization, and there is lot of post PCR processing which is required in order to visualize the PCR product. At the end of it, you are only visualizing the size of the product, but you are not visualizing the number of copies, how many copies, quantitation is not there. Okay, so these are the disadvantages and advantages of real-time PCR. The detection is capable of a very sensitive concentration, and it is going through the exponential growth phase. Then the amount of fluorescent signal is directly proportional to the amplicons generated, and then the probe provides a record of amplification of the amplicon. So these are the real-time advantages of the real-time PCR. Okay, so now we have come across the necessities, or we have seen what are the pros and cons of doing a PCR reaction, whether you should choose a quantitative PCR or you should choose a qualitative PCR. That depends upon our choice. And now, once we have decided to go ahead with PCR, you require to design primers. What is a primer? Anyone from the participants can tell me what a primer is? Any of the participants can tell what a primer is? Participants are there or slept off?
Short segment of RNA.
So it is a small piece of how many nucleotides extending up to 10 to 20 nucleotides which are there, and how to design or how to select those primers, and from which region of this gene you have to select the primers, and what is the sequence which you have to identify for this primer. So design of the primers for the PCR is a first requirement. The other parameters like taking the enzyme, all these are commercially available, but once we design a PCR primer, we need to get the primer synthesized. So we need to be very sure what are the primers which we want to get the primer synthesized. So what are the general requirements? This I'm giving a very abstract information. So depending on your external experimental conditions, all these parameters would change. This is just an overview or a brief covering of the requirements. So we have two important aspects here. One is the general requirement that is related to the melting temperatures. How much should be the melting temperatures? What should be the GC content? And it should also avoid self-complementarity and also the hairpin structure, so that the primer is not going to get amplified, but it is the gene of interest which will be amplified due to the primer. So that is a general requirement while designing the primer. Additional requirements would be in case of a reverse RT-PCR, which is reverse transcription, the primer should be located near the exon-exon junction, so that we are looking at the exons itself, that is the coding portion and not the non-coding portion. And another point is, once we have designed a primer and you are testing it on a DNA sample, and that DNA sample is having some SNP in the site where you have designed the primer. So these regions, which are the most common regions where SNP can occur, it is better to pick primers outside of such regions. So this comes only after looking through several sequences and understanding what could be the possible site where an SNP can come, so that you can avoid picking the primers for that region. And target specificity. To achieve these goals, we have some software tools which are going to help us in aiding the primer design process. And we also have another program which is called as the Primer Blast. So the Primer Blast is another variation of in silico tool in order to design the primers. So most widely used primer designing program which is available, I will be showing that to you also shortly. That is the Primer 3 programs. So but the Primer 3 program is not specifically the target analysis. It uses the typically have to test the primer specificity using additional tools. If you provide a sequence, it will design the primers, because there we are not selecting whether you are taking a gene intron region or an exon region. Whatever selection or whatever gene sequence you give, it is going to design the primer. This process is time consuming because you need to check back whether you have really working with an exon region or an intron region. And these programs, other programs which are useful in designing include an in silico PCR that is there at the UCSC site, which you just seen with respect to chromosome gene sequences. We will be seeing how to design the PCR primers in the in silico PCR and reverse ePCR. They do not design primers, but they determine the amplification targets of the user supplied primer pairs. So the fundamental difference in a primer blast and a in silico PCR and a reverse PCR is that primer blast is directly looking at the sequences and giving us the primers, whereas in silico PCR or ePCR, we give the primers and it amplifies the sequence and shows out the output. Okay. So let me just come out of this and now move on to the basic tools. Now we have seen that there is 3 programs I have given you. One is the in silico PCR, another is the Primer 3 program, and another is the Primer Blast. So let us see each of this for a brief point of time. So if you are able to do it along, or you can do it later when you have completed the class, or right now you can follow now and then do it later. Okay. So let me first open the site.
Ma'am, your screen is not sharing.
Yeah, I'm just sharing. I just stopped sharing because I want to use the browser, so I stopped sharing the screen. Okay, so I'll go to this place which we are all familiar here. Okay, now I will show you the basic simple program which all of us can be use. All are simple to use. This is most user friendly. So what I have done is I have gone to the browser. I have selected Primer 3, and so when we want to predict the primers or design the primers, we take the Primer 3 tools. There are several other tools. If you scroll down, you will find many other primer tools.
Padma ma'am,
Yeah,
screen sharing chesara?
చేశాను అవ్వట్లేదా?
No, we are not able to see. కాలేదు ఇంకా.
Now
Now it is visible.
Okay, so what I have done is I have gone to the browser. Here I have put it as primer 3, and then we are selecting the primer 3 here. So now you can see that there is this is the way the screen appears for us. What is it that we have in this screen? Here you have a window which is there. So here you have, you can include the FASTA format of the sequence which is there, which we all are familiar here. So how do you get the FASTA format? To get the FASTA format, we need to go back to NCBI site. Go back to the NCBI site and go to the nucleotide. And here we can give some sequence. I'm just giving trypsin as an example here, selecting some name here. And this is having a large number of sequences which are here. You can see that this particular thing, small file is too. Okay, so I have given this trypsin I'm selecting here. So I'm taking a small page. For example, here there is a small size of DNA which is there. So I'm just taking this FASTA sequence. Copy the FASTA sequence, or you can also send it to file and upload the file. That option also is there. You can copy the sequence, paste it in this window, and you can remove the other. So this is the FASTA sequence, making sure that this particular line is there. So here it is a pick left primer, pick right primer, and then we are not changing any other parameters here. You have the minimum primer size of 18 and optimum size is 20, maximum is 23. These are the general primer picking conditions. As I have told you in the PPT also, there are some specific conditions which you have to see. Melting temperature, primary melting temperature, that is 57 and 59. We are not changing any of these. GC content, so should be minimum of this, maximum it should not go above 70. And because of the GC content is more, then the melting temperature also will be increasing. So without changing any of these parameters, you can see that there are many conditions which are required here. Salt correction is there, concentration of DNTPs, how much concentration you would like to put. All these parameters are there. And then these are by default, all these values will be there. Then we would be clicking on pick the primers. So you can see that it has taken the sequence and it is giving out output. You can see this is the arrows which are given here. These are the forward arrows, and here you have the reverse sequence. So it has identified or given primers for all in this entire sequence without us specifying what is the region where we would like to pick the primers, or which region in this entire sequence we would like to amplify, because there are so many bases. So which region of this we would like to amplify, and why only this region we would amplify, we have no reason here. So this particular program has taken the sequence and it has randomly designed or given the sequences primers. And as I have told you, there should not be any hairpin. You can see that there is this content hairpin sequences are 0 here. GC content is 50% and melting temperature is also optimal melting temperature. As we have seen, it should be between 30 to 70, and it is well within our range of 30 to 70. 59 and 58 is the optimal temperature, and more importantly, it should be around 55. That would be the most ideal melting temperature which is there. So it has taken the sequence from 272 position and ended up at the 441 position. And so this is the most suitable primers which can be used. So if you're going by these primers, what is it that you can do is we can use these primers and get the primer synthesized by the manufacturer or whoever is making these primers. So we need to be sure when we are doing this, whether we are taking the whole gene or we are taking a particular exon or what is it that we are interested, we should be careful while picking the primers. And here there are other examples, other primers also which are given. There are 4 primers which are offered here. So you can see what is the difference, why 4 primers are offered here. The 4 primers, they may be differing with respect to their GC content. Here you can see it is 50%, this is 55%, this is 50% and so on. So there will be some difference with respect to the characteristics of these set of primers. And these are the additional oligos. If you are still interested to check any of these also, you can get the oligos synthesized as per this sequence order. So this, as we have given an upper limit of 20 nucleotides, this would be containing somewhere near about that. And you can, this is one of the simplest methods which you are using the primer. So you can see the sequence size is 900 and this is included region also is 900, but whether this region is an intronic region, which is the intron or which is the exon, we have taken the DNA sequence, so we do not know whether it is a intronic region or exonic region. Okay, so that is the primer 3 program or primer 3 output, what we are looking at. Okay, so now there is another source where we are going to be looking at, that is this is called as the insilico PCR. Insilico PCR. So if you just click or go to your browser and type as insilico PCR, you will be getting 2 resources here. One is the UCSC genome browser, which we have just now, you have been shown by Dr. Vanita. And you have another site, which is the insilico PCR amplification. I'm just opening both these sites in 2 different windows just to make you understand what are these tools. Okay, so I hope the screen is sharing.
Yes, ma'am.
Yes, ma'am.
Yeah, so this is the UCSC browser which is there. This is one window where we can do the insilico PCR, and here you have another window. This is basically what we are not here. It is not concerned with the human sequences, whereas here you can basically work with many mammalian sequences, because once you go to the genome browser, in this genome browser, genomes, you have these different sets of genomes where you can see that you have already been shown in the previous session. So we have the genomes related to all this different species: human, mouse, rat, zebrafish. Those people, those who are interested in a specific model organism, you're interested in Homo sapiens or mouse or rat or zebrafish, or these are all the model organisms. So you can, if you select any of these organisms, you will be getting the reference genomes here. So we will be looking into the humans. So in the humans also, we will be more comfortable or looking at this HG19, and we will be looking at the HG19 sequence browser here, and that is this is the one which you can use if you want to use insilico PCR for the human genome sequences or any other sequence. If you are interested, the person's if your model organism is not any mammalian species, but it is some bacteria, any prokaryotic genome, this is the site which is here. This is the site where you have a list of species which are there, and These have been arranged in alphabetical order. If you are interested to, if you know the name of the organism, you can, if you want Acinetobacter or Acetobacter, you can select Acetobacter and you can move further here. But what is important here is you have to give the particular sequence. Okay, you need to put the sequence in this window. So how to get the sequence in this window means you need to go to repeat the previous steps what I have shown you. To choose your gene of interest in that organism, first identify the gene which is there in that particular organism, and then that gene you have to put it into your primer 3 program, and the primer 3 program will design the primers whatever we have seen in the primer 3 program. Okay, anyway, so in the primer 3 program, we have seen the sequences. I have shown you the primers. So that we cannot paste it here because those are for a different species. So we can't use those primers. You have to see the gene in your organism. What is the gene you are going to do? And for that, you take out the FASTA sequence, put it into the primer 3 program, and then pick up the primers from that site and paste the primers in this window, that is primer 1, primer 2. And here after placing the primers, you can select if your species is not here, any of your genus, Acetobacter genus is there, but this species you are not interested, or you want to apply for all the species of Acetobacter, you can select on apply to Acetobacter, and then you can click on amplify, which is going to amplify the sequence, and it will show you on the screen what are all the variants which are there, what are all the various species which will have amplification of that particular gene. Okay. So let us, that is one way by which we can amplify. So this also, what are you doing? You are giving the primer input. You are not asking the system to design the primers on the gene sequence, but you are yourself looking at the gene sequence, looking at the nucleotides, and you are asking this tool to pick up the primer. So it has picked up the primer. So that is a way this in silico PCR for this microorganism works. Now let us go back to our this genome sequences here. Coming back to the UCSC genome browser. So in this UPSC genome browser, you have some tracks which are here. These tracks we are not interested would work with all these tracks. If you don't want to work with the tracks, you can keep the hide all button, close it, and all the tracks will be hidden. But today we are going to see how to use the in silico PCR. So I'm just trying to give an example here. Any particular gene, for instance, I will give a tyrosinase gene. So this tyrosinase gene, once you enter your name of the gene, from where will we get the gene? Again, the source is that you go to the NCBI site, you type the gene name, get the correct gene nomenclature and put it into this window. And once you see that, you know that previously it was on chromosome 22. Now it has come to chromosome 11. So this chromosome 11 is the chromosome where this particular tyrosine gene is located. So here we have some selection. We need to take the UCSC genes. All the UCSC genes are there. So in the previous versions of this UCSC browser, you had UCSC genes, but now in the latest version, you have this particular example, that is the old UCSC. So this has to be under, it is already selected to pack. Okay, so this is the option selection which you have to do here. And then we need to look for the amplification of the genes. So here we will go to the view button which is there on your scroll bar here on the top. view window is there, and we will click on the DNA sequence. We will click on the DNA sequence, and we have the extended case color options. So why we are using the case color options is we would like to see where is the exon region, where is the intron region. So in order to differentiate the exons and the introns, we are selecting this particular case, default case, upper and the lower case. And here we will select this because already in this current one it is there. The UCSC genes, this box has to be selected here by default because I used it in the earlier version. So this is selected. Otherwise, on your screens, if it is not selected, you have to put it click box on this, and here you can choose the color of the sequences which you would like to see. That is red, green, blue is there. So you can change the number here to any color, and you can in this column you can change the now you want red color, you can choose red or green or blue. So this is what it is, and then I'm going to click on the submit. So now you see that there is a differentiation which has come up here. So there is some sequence which is important here, and down if you scroll down also there will be a particular segment which is there. So which means that all these highlighted regions could be the possible exons. So we are looking at these regions on the UCSC browser. So I'll just go to the presentation for a minute. Is my presentation sharing?
Yes, ma'am.
Yeah,
Yes,
so
ma'am.
here you can see this is the one which I have shown you. And this is the way whatever we are going to do. We need to check this assembly, then check the color options, and then you will be getting this sequence and like this. And this sequence from here, you have to select the nucleotides from the top portion and from the bottom portion of this same sequence. So here you can see what I'm trying to say is we will be doing only for the exon 1. So this is exon 1 and this is exon 2. So we are not taking the rest of these nucleotide positions here. Then so we need to select this particular sequence of the first exon for the forward primer, and the sequence from the end of the first exon. This is the end of the first exon. We need to select the primer for this. And then we have to select the insilico PCR tool from the toolbox, and then it would do the amplification of the gene. So let me see if I am able to show you that here. If we are not able to do, you can try it out with the different genes. Okay, so here I have this is the sequences which are here. So what I am trying to tell you, as we have seen in the previous slide in the PPT, we are going to take first few nucleotides from the beginning and the end of the nucleotides. Okay, so what we have to do is to go to the tools. Okay, and now before going to the tools, what we have to do is once you select this sequence, you can select a small segment of this sequence here, and point your cursor and drag it, it will get selected. As you do any copy paste action, this also you can do the copy this set of sequence and paste it in a window, separate window. I am pasting it in the Word. You can open a text box, or you can open a notepad or whichever text document you have. You open that and place the sequence. That is going to be you can label that as your forward primer, forward sequence. For the forward region, you are going to label this. And coming back to this, as I have shown you in this slide, we will be taking the last part of the sequence. I have shown you we'll be taking the sequence here. So we will go back to our window and select a last portion of this segment from this region. Again, click on copy, go to the Word document, paste the sequences here. So you can see that this, I have selected slightly shorter sequence. So maybe we can remove this from here and make it uniform. So now these sequences are here. Now we need to go to the insilico PCR tool, which is here on this toolbox. In this, you have the blue bar on the top of the window. There you have the tools. There you can select insilico PCR. This will be opening up here. Here you can go to your Word document and go to the Word document, copy paste the sequence. Come back to your UCSC browser and paste it here. And here take the reverse primer. Take the reverse primer, copy the reverse primer and paste it in this sequence and make sure that this particular selection is there. Flip reverse primer so that you are making sure that it is there. Sequence is a little shorter. Anyway, so this is what we are going to do. We are not changing anything here. You can, maximum product size is 4,000. Okay, so this is what we are going to work it out, and we will have to, we'll just want to check this primer once again. Yeah, it is there. So click on the submit button. Click on the submit button. Okay, once I click on the submit button, you can see that we are getting an output here. This is the output. It is having around 892 base pairs. This is the region which is taken up, amplified for this particular region. So there are 892 base pairs is the size of this particular amplified fragment. And if you want to see this particular fragment which is amplified, you can click on this and it will tell you the region which has been amplified in this. It will show you where your gene is. This is here says your sequence from the PCR search. So this is the sequence which it is going to amplify, and it is on the tyrosine gene. You can see. Is it clear to all?
Yes, ma'am.
Could you follow this?
Excuse me. Can you explain one step extended DNA case and color options?
Yeah. So here, when we select this sequence, you go to view portion, get your DNA sequence, provided you have selected, and then here you have the extended case color options window is there. Here you need to select the highlight the regions which are important. Okay, so for that you have to be selecting this box has to be active first. UCSC genes has to be active. Along with this, in this version of the UCSC, this gen code has been newly added. In the previous versions, there was only the UCSC. So now this gen code has been added. This numbering is, which is here, this is only telling us the size. You can make it increasing the size of the letters or whatever it may be. And you are increasing the color. You are choosing a blue color, or you can, if you don't want blue, you can keep this 0. You want to see a green color, you can select the green color. Anything you can select here and click on the submit button. So if you select like that, it will show you the green color there. Because you have selected green, it will select green. If you select red, it will select the show you the, because we want to see it more clearly which regions we are amplifying. So we will be doing this.
Okay, ma'am. Thank you, ma'am.
Yeah, so that is, and finally, that will be the output which we are getting here. So this is the output which we have got. I have already shown in the window. So this is the output which we would be getting when we do the in silico PCR. So in this in silico PCR, we are trying to amplify the sequence and know the size of the sequence also, which we can, when you do the wet lab PCR, we don't know the fragment, whether it is amplified, whether the size is amplified, whether these primers are really amplifying that sequence, and what are the regions which are going to be amplified. The position of the sequence is not known, right? So this position or this particular tool will tell us the indication, and also you can go and check back whether the you can also go and check back whether this gene sequence is really amplified or not. Okay. Okay, now again let us go back to the usage. So that is, I have shown you two tools. One is for the microorganisms. What is it that you are microorganisms? If you want to amplify the sequence, you need to go to the gene ID, pick up the sequence, FASTA sequence, go to the primer 3 program, paste the FASTA sequence in the primer 3 program. It will give you the primers. Use those primers and do the amplification of those genes which are there in the by using the in silico PCR site of this particular site. This is the URL insilico.ehu.es. This is the URL. Maybe I will just copy that and try to see whether I can put it in the Okay, so this is the URL which is there, insilico.ehu.es. So there you can use the sequences which are related to the microorganisms. So I think I'm running short of time. So I quickly go through this, whatever I can show about the other tool which I wanted to show. That is called as the Primer Blast. So for the primer blast, this primer blast tool, you can access it. You need to specify the range from where you would like to select the primers, or in which region you would like to select the primers. So in order to do this, what you can do is go to the NCBI site. My screen is sharing.
Yes, ma'am.
Yeah, go to the NCBI site and go to the gene link. Go to gene. Here I'm just giving some example like trypsin. Okay, now you will be getting this particular window. So here you can see if there is all these are different species. We can select maybe Homo sapiens here to make our work easy. Because why I'm insisting on Homo sapiens is we are going to see in the UCSC site. So this is not showing. Okay, so this is Xenopus. Okay, anyway, so here this particular one window, I have taken an example of a gene, and the gene which I have taken is a trypsin from this particular species. Now what is come down here, you can see this particular window here. You have these lines which are here and the solid bar which is highlighted here. So these are, if you put your cursor or put your cursor there, it is going to tell you that this belongs to the exon. It is an exonic region which is there, and this particular region is a region which is between the two exons which is there. So we would like to amplify, for instance, a particular region of this exon. We would like to amplify this exon. So this is exon 2 of 5. You can see it is written exon 2 of 5 here. This is exon 1. If you just hover around that, you will be getting exon 1 of 5, and this is exon 2 of 5, and so on. So now if you want to amplify this particular exon which is here, now you know that you are amplifying only this particular exon, and you would like to amplify this region without losing any portion of the gene. So what you can do is you can move on to this solid bar which is there on the top of this window, where you can see the position numbers are written here, 501 and so on. So you have to get this line. I hope you are able to see the line which is there. So when the line appears, I'm going to click on my mouse and cover it like this, so that I'm taking this particular sequence, and then I'm going to click on the shift button in the keypad and come over to the other side of this exon and select this portion and this portion. Okay, when I select this portion, there are some options. When you just select this portions, you will be getting some options here. That is, you can select the primer blast. So this primer blast, now it has taken this region and this region. So now what we have to do is go back here. Since it is not having the forward and the reverse one, what we have to do here, so select a particular segment here and hold the shift . Hold the control button, not the shift button. Hold the control button and select this, and then you will be getting the blast, primer blast here. So now you can see that 500,549 to 644, 872 to 500,999. So these are the position of the forward primer. This is the position of the reverse primer. For this accession number, automatically it has taken up here, and rest of the parameters we don't need to change anything in this, and then we can get the primers. Right, it will take a few seconds for the primers to be loaded up, and then it would give you the output. Now, what is the advantage we are getting here? Advantage what we are getting here is we have really seen the region where you are amplifying the sequence, instead of taking the whole sequence, putting it into that and looking at whether it is amplifying the required sequence or not. And moreover, if you want to work with only a specific exon, if you know that an exon is having an SNP, that is a single nucleotide polymorphism, you want to work with a gene, disease gene in case of human cells, you want to work with a disease gene, and you know that that particular exon is having an SNP, so you can design the primers. Instead of designing for the whole thing, you can design only for the specific DNA, specific exonic region. Okay, so let me wait for a little while. Meanwhile, if it comes, you can see it is still making. So it is saying making primer specific to the template. Maybe it will take some time. So that is, once it is done, if it's done, we will again go back and check. So that is what I would like to show you today. I hope it was useful to all the participants, how to design the primers, how to identify the region, and how to do the in silico PCR. Any questions, participants? Exhausted?
I'm having a doubt, ma'am.
Yeah.
I'm
Tell me.
getting options differently in this one. Can I
Yes.
share the?
I will stop sharing. You can share yours. Yeah, you can share your screen now. Showing, are you sharing the Arina? Maybe you can send a screenshot to us on WhatsApp. We can, yeah.
These options it is showing, ma'am. Can you see?
Just a second. Which is this? Where is this? In extended DNA case color options. So many options are coming for you.
You see that one I'm not getting, ma'am.
You go back one step. Did you select human genome or you select something else? Yeah, get DNA.
It is human genome, ma'am.
Yeah. Chromosome 7 you have taken, is it?
Yes, ma'am.
Just a second. So here, what is it that you have taken? The sequence formatting options and all are coming. I think somewhere you did a wrong selection, maybe. That all uppercase, you take it out. Is it going that all uppercase sequence formatting options?
No, ma'am, it's not going.
Okay. I think we need to check your browser. Maybe you can try it once again with whatever I have shown. Once you can get back tomorrow again, we can look into it. Okay.
Okay, ma'am.
Let me see if this is done. Yeah. I'll just share the screen. Have patience for another 5 minutes. Yeah. So is the screen sharing?
Yes, ma'am.
Yeah. So this is the primer blast which has been done, and it is showing the template region from where we have seen, and this is the region which it is going to do the amplification here, and it also is giving us the primer pair which is important here. Product length is 374 and you have this sequence length which is given here, because that depends on what is that region we have selected here. Along with that, it is also giving, see for instance, here the GC content is 75% and the melting temperature is 60%. And along with it, it is also giving some other templates which also could be probable templates for the amplification of the PCR. So this is how the primer blast is going to, this is the primer pair 2 which is given here. This primer pair 2 is having around 383 product length. So you choose the best out of this looking at the TM and the GC conditions. So that is how the primer 3 output, the blast output is giving you the most common or the most relevant primer on the top, and other primers, optional primers also would be given in the bottom if you scroll down the window. So that's how we can generate primers and also do the PCR amplification of the genes of our interest. So that's all from my side.
Okay, thank you, Padma ma'am, for giving a detailed explanation about in silico PCR and primer designing. So I thank both the speakers for today's sessions, Dr. S. Vanitha ma'am, regarding the gene analysis using Genscan, as well as Dr. S. Padma for the in silico PCR and primer designing. Now I request Vanitha ma'am to post the attendance form. Ma'am.
I have posted, maybe participants can check it out.
Participants, please check if you can have access or not.
Yeah, please check it out. And any queries are there, you can stay back. Others, after filling the form, you can leave. Padma, is that okay?
Yeah, fine for me, Vanita. If they have any queries, we can take up.
Yeah, they can stay back.
Or you can also send it through WhatsApp in the group. You can see if anything can be done on it. If you are willing to stay back, we can stay back and solve the issues. Others can complete the form and leave the class.
Hope all are able to fill the attendance.
Yes, ma'am, they are filling.
Okay.
Number is increasing. They are filling.
Excuse me, ma'am.
Yes, ma'am.
Ma'am, actually, I'm unable to look at the form in the chat box. Can you send it again?
I'll do it now. Just hold on.
Ma'am, if it's possible, could you please send at least the notes of the class, ma'am? We can refer back to the notes and practice the whole thing once again.
Yeah, we are going to share it in tomorrow. By tomorrow, at least the initial classes, what we have completed, the PPT, we will share. We'll start sharing it to you. We will do it.
Yes, ma'am. Thank you, ma'am.
Recording you have stopped, ma'am, Vanitha ma'am.
Yes, ma'am.